Hello everyone, this is Ph.D. Daddy. Today, as a special feature of the 2022 Nobel Prize in Physiology or Medicine, let's study what Dr. Svante Pääbo has studied. Surprisingly, he is the only winner unlike usual year. First of all, I would like to express my sincere congratulations and respect for receiving the Nobel Prize. He was born in Sweden. He is currently doing research at the Max Planck Institute in Germany. He is a researcher who studies the evolution of mankind using the DNA of ancient humans. Are you guys wondering how to study human evolution with DNA sequences? Then let me briefly explain the principle. Let's say a person's DNA sequence is AAAAGGGG, and this man gave birth to a child. However, due to a mutation, this child's DNA sequence became AGAAGGGG. Again, this child grew up and gave birth to a child, but a mutation occurred in another region in the DNA. And this baby has the DNA sequence AGAAGGGA. As you can see, DNA mutations are accumulating with each generation, right? Then, if the fossils of these three people are discovered in the distant future, it will be possible to determine who is the grandfather, son, or grandson through DNA mutation accumulation comparison. However, DNA mutation is not a very common event. According to research, every generation passes, 1 or 2 mutations out of 100 million nucleotides occur. Now that we know the frequency of DNA mutations, DNA mutation accumulation studies allow us to know how different fossils are genetically, how long it takes for that gene accumulation, and when they diverged. However, there is a problem. Ancient human fossils are so old that only trace amounts of DNA remain. And the DNA that remained was also chemically denatured and mixed with the DNA of microorganisms, making research difficult. Dr. Svante Pääbo has developed a method for stably extracting DNA from ancient human fossils after a long period of hard work. So let's take a look at how to extract DNA from ancient humans. First of all, the experiment is being conducted in a separate space. The researcher is wearing a protective suit to prevent DNA cross-contamination. And they use UV to sterilize all external microorganisms. Usually, when we extract DNA from tissue, researchers are not that picky like this. Using this extracted DNA, the professor was able to continue his research on DNA mutation accumulation. And in 2010, he surprised the world by discovering a previously unreported new hominin called Denisovan. He also found that modern humans, Homo sapiens, mated with Neanderthals and Denisova. So he also studied the effect of genes from Neanderthal on our immune system. Because of his research, we also learned that the cause of our vulnerability to the COVID virus is due to genes from Neanderthal people. Professor Pääbo's research, who broadened our perspective with the study of ancient human DNA. Isn't it wonderful? Once again, I would like to express my gratitude and congratulations to Professor Svante Pääbo, who has been walking the path of research for a long time. Then we'll see you in the next contents.